At the Global Liver Institute, we believe it all starts with a patient. So what happens when a patient, a liver patient no less, one of the 500 million people living with conditions that often go undiagnosed, untreated, or misdiagnosed, mistreated, and stigmatized, brings the challenges of liver patients front and center? And what happens when a patient then leads a discussion with world experts in policy, research, clinical care, and wellness on ways to overcome these challenges? Let's find out today on GLI Live. Hello and welcome. My name is Donna Cryer. I am the CEO of the Global Liver Institute, and I have the privilege of being your host for this episode of GLI Live, which is part of our series this month for Women's History Month, to show to highlight and to spotlight women who are making history, particularly to showcase women who are succeeding in the sciences. Today, I have with me the director, founder, chief medical officer of Madrigal Pharmaceuticals, one of the innovators in hepatology in the liver health space, who we are so excited to partner with at the Global Liver Institute. Welcome, Dr. Taub.
Hi, Donna, and please call me Becky.
Thank you so much.
Happy to be here.
So it's always wonderful to me. As a young girl, I was on the chemistry. I was the only woman on the chemistry team, the only girl on the chemistry team. And yet I didn't really feel like a career in the sciences was something that was meant for me. So can you share with our audience how you got interested in science and did decide to continue on this path?
Yes, so I was interested in more in science, math, way back when I was in college, which was a long time ago. And I actually originally did a lot of work in laboratory-based works like biochemistry, essentially, but decided that the field of medicine would be better for me because I like the fact that it was a much larger environment of people that you would interact with than, just a research lab, or which would be, basic research. So I was interested in research, but I also wanted to have interactions with patients and all the other people that were providing medical care. So that's why I ended up going to medical school,
Mm-hmm.
and doing a residency in internal medicine. And then when I was in about my third year residency, I realized that there were so many diseases that we didn't have treatments for, and seeing young people sometimes dying of really horrific diseases and things like that, and there was a limit to what I could do as a practicing physician. So I ended up doing a genetics fellowship at the NIH and then at Harvard Medical School. And we focused then on cancer research. And then I ultimately ended up in the genetics department at the University of Pennsylvania as a Howard Hughes investigator. And that's when I started working on genetic systems in the liver and working specifically on liver regeneration. So it was kind of going from science to medicine and then back to science again, but with a more medical application. And I was at Penn for a number of years, and until about a little over 20 years ago. And I was a professor of genetics and medicine at Penn, and I had done a lot of work on liver biology, and I decided to enter pharmaceutical research as sort of a change in career focus, with it's more applied, obviously, than doing basic research in an academic institution.
Mm-hmm.
I was a researcher and a teacher
Mm-hmm.
and all of that. And so ultimately, it evolved into Madrigal. But there was a ways to go before we got to that point. So
that is quite a journey. Uh, my husband also made that leap from the CHOP genetics department into industry, and so I know the quality and the rigor of that department, really singular in the country in many ways. So you went into industry first in large pharma. What were you telling yourself as you made the leap into your founding your own company?
yeah, so I when I left Penn originally, it was actually to join what was called a medium sized company.
Mm-hmm.
It was a division of DuPont,
Mm-hmm.
and it was called DuPont Pharmaceuticals, and it wasn't that large, but it was
Mm-hmm.
actually an incredibly good learning environment for
Mm-hmm.
how to discover drugs and develop drugs. And then we were bought by Bristol Myers Squibb, so
Mm-hmm.
I didn't choose to go into large and I think that's really kind of the message that um, uh, at that time it was kind of what happens in the company environment that's
Mm-hmm.
different from an academic environment is that you don't always know what's going to happen and what the
Mm-hmm.
various transitions that might occur. So I was at BMS for a couple years and in different leadership roles. So while I was at DuPont, I headed up a metabolic disease group,
Mm-hmm.
but also CNS, central nervous system
Mm-hmm.
research, because we were doing very genetic based research. We actually, our obesity work was within CNS, and we had programs in Alzheimer's. So it was really kind of a transition almost from genetics at Penn. And then after being at BMS for a couple of years in those sorts of roles, I ended up at Roche, where I was the VP of the metabolic disease research department, and that's where resmetirom, which is the molecule that Madrigal is developing
Mm-hmm.
for NASH, was actually discovered. Was in that
Mm-hmm.
department at Roche. So the transition from Roche to the small company came about again because of a transition within Roche itself, within the company, where they reorganized some of their therapy areas, and the one that I was involved with was moving its headquarters to Basel, Switzerland, and that wasn't something that I wanted to do at that time, having kids in high school, et cetera, back in Pennsylvania. So I ended up getting a really nice offer from Roche to be able to license for very little the program that became resmetirom at the molecule and the patents associated with it, and then one other of the programs from our group at Roche.
Mm-hmm.
And that's how Madrigal eventually started, was based on those Roche assets.
I think that sort of that licensing and spinning off and tech transfer and repurposing of drugs is often something that patients and the public don't realize is going on, and that it's often a way that a molecule or an asset which might not fit the strategy of one company, but still has promise. And it's a different way that we can support research to support those who want to move those assets and continue studying something in on its own in a smaller startup or biotech. It's something I've learned to watch from seeing the early days of your and talking to your early medical advisors, and was always fascinating for me as someone who's, as an advocate, is trying to find a way to support innovation in the field, as you said, for so many of the diseases that we have, including my own, that don't have treatments. And so understanding this ecosystem is a way that we can be more supportive of research. And so one of the questions I want to ask was, what surprised you most about going off and creating this on your own? You had already been successful in so many different other roles, and I'm assuming learned so many different things. What was surprising to you, or what was your biggest sort of aha moment as you sort of founded this?
Well, I mean, I would say that I wasn't really very good at it. I wasn't in the sense of not being really business oriented, because I'm not.
Okay.
And so it was the support of the venture capitalists and other people that were financing the effort, and they saw the opportunity that we had with the programs that we were able to license, and took care of that part of it. I think I certainly learned a lot about what it does take, to raise money and start a business, et cetera. And I think everyone gets surprised by that, because if you're trained as a scientist or a physician or whatever, it's not a natural thing necessarily to go out and try to raise money and speak to investors. Very used to certainly talking about your science and your program and all of that, but having people engage in that opportunity is a whole other thing. The other thing that I think is a part of a transition from big pharma that you mentioned, which can be a difficult transition, is that big pharma tends to be very self-contained. So all of those different types of functions that you need in a small company, that you need to operate very virtually and have work with many vendors and people that conduct activities. So if you wanted to do research, it's not like you just walk into your lab like you would at Roche. You have to find someone outside who's going to conduct that research, and you oversee them from a distance. And so you have to establish this network of CROs and regulatory people and chemical people and all kinds of different functions that are part of a big pharma company. But as a biotech, you have to go outside and find them. And so you develop a very good network. I don't think big pharma prepares you very well to be able to do that. I think actually having been worked in a more entrepreneurial environment as a Penn professor was better preparation for something like that. Where you're used to creating networks with other collaborators. So that was an important thing we did over the first few years after Madrigal was founded.
I think you also said something that's very important for our listeners, that you didn't have to know everything at the beginning. You didn't have to you could rely on a team. You were outsourcing things. You were building your network. I think many people, particularly many women, feel that they have to sort of know everything before they can start. And that's, simply not the case. Sometimes I will point the camera down to this large stack of, black and yellow XYZ for Dummies books that I have. I'm, constantly like learning new things. I'm like, I didn't know that when I started, but I wanted to start. And then, learn things along the way and ask people questions and, got in some advisors, formal or informal. And so I think that the most important thing is to start, not to think that you have to know all the parts of certainly everything before you begin. So I think that that's really important.
Yeah, I mean, there's no way you could possibly know all the parts of what you have to do, because there are so many different aspects of, I mean, coming up with especially in this field. I mean, I think that's true of science in general, of course, but especially if you're trying to conduct drug discovery or drug development. I mean, there were so many different functions, and you do have to rely on outside people, and it's something that you can really get tripped up if you don't have the right expertise in a particular area. So yeah, absolutely.
So, in the time that you've been building Madrigal, the liver space has changed. There were a lot more companies in the space. There are a lot more clinical trials on many different liver diseases. How has that sort of growth or changes in the space affected your company and just sort of how you see yourself operating?
So I think you're absolutely right. There's been a big explosion in the NASH area. I mean, when we first licensed our assets from Roche, we knew that resmetirom had clear activity in the liver, and it also has it's very, very active in lipid lowering, which is part of the same activity, but also a unique activity. And there was no development path for NASH. So our early studies were focused more on the lipid lowering properties of the drug, with NASH kind of in the background. And I remember thinking, oh my God, I don't want to do a liver biopsy study. I mean, like, that just seems, very, very difficult. And it was difficult. But it was really at that time, and I don't know what your own personal interaction was with what was going on, but it was not that many years ago when we first got the FDA interested in potentially coming up with a development path for a NASH indication. And there was some early data from the NIH and Intercept related to obeticholic acid. And I think that was the trigger people to start looking at, well, do I have a drug candidate that might work in NASH? We had an advantage because we had looked at the liver very carefully, our early studies. And I think in some ways the fact that I'd worked on the liver for so many years at Penn actually that. So we got moving early, and we had one of the first handful of phase 2s that was conducted, and we had far fewer tools at that time. So that was sort of the 2016 to 2018 time period. About five years ago when we finished that, we had far fewer tools than we do now. So there really has been an explosion, especially in the diagnostics, the use of FibroScans. I remember buy fiber scans for a few of our sites that were very effective and accurate. We had MRI technology, which we used in our study, and some other serum biomarkers. But the amount of information and publications behind that was very limited, and it's been an explosion. So I'd say on the diagnostic side, especially, there's been so much work done in the last 5 or 6 years, as well as obviously thousands and thousands of different NASH biopsy patients have been assessed, when prior to that it was really just a few hundred patients who had been studied. I mean, the literature was quite limited, as you're probably aware.
Yes. You asked my relationship at that time. So I was probably at least 6 liver biopsies in personally. And professionally, I was the patient representative on the FDA AdCom for obeticholic acid. And so having the opportunity to sort of read everything from that side and that point, really had me invested in the regulatory science and pathways to be able to support, meeting serious unmet needs in the liver space. So it's been great to watch on both sides, and having recently finally gotten a fiber scan, I'm even now more encouraged about what we can do in non-invasive diagnostics. I think really, certainly in the research, and to have more patients participate more easily in clinical research, the non-invasive diagnostics will move things forward. But I see, and I would love your perspective on the growing gap that we see because of in clinical management of patients. Non-invasive diagnostics have wide adoption and acceptance. In the research space, we're still using biopsies, and I have these these images that at some point they're gonna clash. And so how can we solve that gap and bring research a little closer to how non-invasive diagnostics are actually widely used now in clinical management?
Yeah, so there is still a gap, in the sense that the biopsy is regarded as the gold standard, in the sense of, well, you have a sample, and NASH was always a histologically based diagnosis. As that was how the diagnosis was established. The accuracy of biopsy isn't 100%, but
Right.
it's not also likely to be completely wrong. And the diagnostic tests we have for NASH, with the exception of possibly the MRI PDFF, which is extremely accurate, are not as accurate as maybe some other disease areas. So look at LDL cholesterol or glucose.
Right.
These are extremely accurate. You can diagnose diabetes with hemoglobin A1C,
right.
And you're going to be correct. Or you can diagnose elevated cholesterol. With NASH, we have not gotten there, and it's important to realize that. When that was my biggest surprise, actually, working in the field of NASH, was compared to some other areas I'd worked on clinically, is that it took us two months of screening to figure out if the patient had the disease.
Wow.
With going through the various of and finally arriving at the liver biopsy, where we made we ended up with the decision as to whether or not they had the disease, as opposed to a simple blood test where you knew the day that someone's diabetes was under control or not under control or whatever. So it has gotten much better, and it's approximating accuracy at this point, but it's not 100% there. And so then the question is, how accurate do we have to be, to say that this patient deserves a therapy for NASH? And, what gives us the highest degree of confidence? And I think that, if we start at the more severe disease, like NASH cirrhosis, so this is very advanced, I'm very confident we can diagnose NASH cirrhosis with, the tests that we have, because as you know, certainly the chemistry starts to change with cirrhosis as it gets more progressed. But even prior to that, the FibroScan, the MRE, when you get to that stage, it starts to be you're getting very accurate measurements, because there is significant fibrosis. You can pick it up. So if you back away from, and as I said, the MRI PDFF is very accurate. If you back away from that very advanced stage, absolutely, I believe I do not need a liver biopsy for 100%. You back away from that, and you're somewhat at almost cirrhosis stage. You're still going to be pretty confident, and you can rule out other forms of liver disease. In Madrigal studies, we like to look at metabolic risk factors. We think that, the more metabolic risk factors you have, and then ruling out other liver disease, you're very confident that they have NASH and not some other form of liver disease. And the more metabolic risks they have, diabetes, overweight, obese, insulin resistant, hypertension, dyslipidemia, all that constellation of factors, it's going to be NASH. It's not going to be just steatosis or fatty change in the liver. So I think that our tests and the liver stiffness tests and the other tests are good at picking up that more advanced fibrosis stage. I think when you get to the earlier stage, that's where, the certainty is less. But we'd like to think that with a safe therapy, and these patients have metabolic risks, that our drug, for example, could still be very helpful in them, even if we're not 100% sure that they've got what's called at-risk NASH. I don't know if that answers your question.
I think it does. It's one of the reasons that from the very beginning of the GLI NASH Council, we invited in cardiologists and endocrinologists, and have always taken the approach that we need a solution for every stage, and really thinking holistically about the benefits of therapies in this space. Looking at the whole person health and your overall, metabolic risk, is something, worth aiming for, and that we would get better at the diagnostics and better precision. But it is something that certainly, when we brought the internal medicine folks on board, they were looking for just that one number or that one test, and I was like, well, we don't have that yet, but hold on, this is still important. They were sort of, call us back when you have it. I'm like, no, we still need you now, because you do have these patients in your practice right now, and so you need to be learning about this. So, as we approach International NASH Day in June, it seems like it's coming up so quickly. What would you love to see happen? What would you want to have activated, and more of your colleagues throughout medicine know about how they can support?
Yeah, I think what you said, they have those patients in their practice. It's like, and that's why we started with the clinical aspects. We said, okay, you can identify these patients. I mean, how many patients do you have that fit that profile? And there's a lot of them that fit that profile. So you've already got a significant enrichment there, without one little test, right? You can say, okay, this is the patient that I need to think about, and maybe get a FibroScan on that patient, because they do have, they are diabetic, they are overweight, they probably do have or fatty liver, and and we can do a FibroScan in them, for example, and further enrich. So if it turns out that it doesn't show anything, then they may be okay. So just making them aware that you may not be able to get a simple blood test, to say, I guess, one of the things that always bothers me about this whole referral network is for liver disease, is that people have these massively elevated liver enzymes, and NASH patients, most of them do not have that. So if just getting that message across, I think is really important. And in our clinical trial, I think we had something like 10 or 15% of the confirmed at-risk, very high, fibrosis stage at NASH patients, 10 or 15% had what would be flagged by general practice as significantly elevated liver enzymes, and the rest didn't. So they might have been mildly elevated or really not considered by lab tests to be elevated at all, but they all had that profile.
Right.
The clinical profile that I know that these physicians can recognize very well.
I think that is a really important message as we describe the risk factors for every physician to look for, because it's heartbreaking to me that there are patients. There's certainly those who are not connected to the system, but there's so many who are receiving healthcare today, who have all of the identified risk factors for fatty liver disease or NASH, and aren't, being either referred to a clinical trial or treated in some way. So we'll work together on NASH Day and every day to do a little bit better. And I thank you so much for sharing all of your vast experience. I think that you've given enough advice for at least 10, 12 different careers in healthcare that women can aspire to, and that people can aspire to. Thank you so much.
Thank you.
Please be sure to like this video, subscribe, and share your thoughts and suggestions in the comments below. Join us weekly on Wednesdays at noon Eastern Time for more episodes of GLI Live. Check out our website, globalliver.org, and follow us on social media to learn more about the Global Liver Institute and stay up to date with the latest in liver health. Until next time, stay safe, stay well, and stay connected.